Hello everyone, welcome to Gebze Technical University Department of Bioengineering webinar series. This is our 27th webinar. Our guest for today's webinar is Dr. Pınar OBAKAN YERLİKAYA from Istanbul Kultur University, and the webinar title is Epibrassinolide, not just for plants anymore. Let me introduce our guest shortly. Dr. Pınar OBAKAN YERLİKAYA received her bachelor's degree from Istanbul University, Department of Biology in 2007. She completed her master's degree in Université Paul Sabatier. Then she received her PhD degree from Istanbul University, Department of Biology in 2013. Currently, she works as an associate professor at Istanbul Kultur University, Department of Molecular Biology of Genetics. Dr. YERLİKAYA, the stage is yours.
Thank you very much, first of all, to Professor Barış Binay for inviting me today, and to Batuhan for helping me through all this technical stuff and Teams, etc. So today I will present our, let me first open my share tray. I hope you see all the slides. And so my team is actually focused on a compound called epibrassinolide, which is actually a plant growth regulator molecule. But our team is focused on the effect of this compound on different diseases like cancer and neurodegenerative diseases. And today I will present all our data in vivo, in vitro, and other model organisms such as Xenograft elegans. So epibrassinolide is actually a member of brassinosteroids. So brassinosteroids are plant growth regulators, and to a certain concentration, you can see that epibrassinolide induce plant growth, and also we can see its antioxidation and detoxification effects in literature, in plants, of course. And 2010, Stegoreva and her colleagues in Czechia, they performed some experiments on cancer cells using this compound that I will call it EBR from now on. So EBR was shown by these people as a hormone receptor inhibitor. So although its signaling pathways and signaling properties of EBR is well known in plants, so we do not exactly know how EBR can affect mammalian cells. So although the experiments have showed that EBR can have some anti-cancer activities, so in plants, EBR is bound to its heterodimer receptor located in transmembrane in the plasma membrane. Upon binding to heterodimer receptors, actually represses some repressive proteins like BIN2 and activates some transcription factors like BZR1, 2 to initiate BR-regulated gene expressions, especially those having role in plant growth responses. And in 2020, now we know that brassinosteroids have therapeutic potential against many diseases, including cancer and other diseases that I will mention on. And starting from this point, actually, we ran some blast assays. So if this compound can actually start a signaling pathway in plants, so if we apply this, and if Stegoreva and her colleagues apply to this compound to mammalian cells, which actually proteins can be affected. So these heterodimer receptors actually can act like mammalian kinases. Especially we saw that mitogen activated protein kinases or other important cellular kinases in mammalian cells can be affected from this compound. And one of my PhD students, she now will terminate her PhD at the end of May, and she made some good results. She combined this compound from with different proteins inside cells, especially steroid hormone receptors, such as estrogen receptor, androgen receptor, and retinoic acid receptor. All these receptor proteins are steroid receptors. And EBR, we show that EBR can target these steroid receptors and can inhibit their actions actually. So we, starting from this point, we hypothesize that EBR, due to its steroid-like structure, can easily pass through plasma membrane and can bind steroid receptors inside cells, such as androgen receptor, as I said, estrogen receptor, and can block their activity, because we know that these steroid hormone receptors can translocate to nucleus in order to initiate their target genes, and these target genes especially have role in cancer development, cell proliferation. So we can see this scenario in prostate cancer or breast cancer cells, because they have a high expression profile of these steroid receptors. So at that time we made some in vitro experiments using prostate cancer cell lines, LNCAP and DU145 cancer cells. So these cells actually, one of them, LNCAP, they express androgen receptor, and the other one express a non-functional mutant androgen receptor protein. Interestingly, we get some results that EBR can actually initiate apoptotic response in these two cell lines. So instead of choosing the steroid hormone receptor expressing one, the other cell lines is affected as well. So we start digging other proteins which might be the EBR target protein, and we search like P53 or other tumor suppressor proteins. Maybe they have role, but no matter what we choose, EBR always induces apoptotic cell death in these cells. We also checked some survival pathways like PI3 kinase, MAP kinase signaling. So we always get the same result, that no matter your cell express steroid hormone receptors or not, cells undergo apoptosis, like we use prostate cancer cells, breast cancer cells, colon cancer cells. And therefore we came up with a result that this compound might be a potent apoptotic inducer, but which exact mechanism is under EBR-induced apoptosis? So to dig all the proteins of the cell one by one is very hard way to find one compound's action. For this purpose, we divided the project into three actually. The first part was conducted in vitro cells. The other part was conducted in vivo mouse models, and the last part is conducted in, as I said, model organisms, C. elegans. So in the first part, to understand the EBR action in a more global way, we made a proteomic approach, SCILAC assay. So SCILAC is the acronym of stable isotope labeling by amino acids in cell culture. So this technique, this approach, actually you label your treated cells with non-radioactive isotopes. These isotopes are lysine and arginine amino acids, with more heavier than the normal lysine and arginine amino acids. So in this way, you can easily track your newly synthesized proteins after your compound treatment. So you can see which proteins are made, because lysine and arginine amino acids are usually in protein structures. So you can easily track which proteins are made, which post-translation modifications they have, etc. So for this purpose, you collect your proteins and you mix them, and then you run a 1D STS page gel, and then you run your samples in a LC-MS. And after that, upon getting your results, you validate, of course, your results, your protein expression profiles by immunoblotting. So our results, so this part of the study was performed in Professor Merali lab in Temple University, Philadelphia. So after my PhD, I moved to his lab to learn all these LC-MS thing and other experimental designs. So our data tells us that ER stress, endoplasmic reticulum stress, is mainly the target of EBR. So I was very glad to run these experiments, because otherwise, to understand one compound effect and to say, so you can see every day there's a paper coming up with saying that these compounds are apoptotic, this compound can be a target for, chemotherapeutic applications, etc. But it's a very long and heavy way to understand. So by doing a proteomic approach, you can see what's going on inside cell in terms of all the expressed protein profiles. So our data, and then we published our data saying that many important cellular processes like cell cycle, cell death and survival pathways, molecular transports are mainly affected. But especially protein ubiquitination pathway was very important for us because one protein, calreticulin, which is actually important for protein folding in the inside ER lumen. So we found that calreticulin is go down regulated after EBR treatment. So what is actually calreticulin? Calreticulin in the ER lumen plays a very important role in order to, as I said, protein folding by the help of calnexin. So these calnexin and calreticulin proteins are actually chaperone proteins, and they try to fold properly, especially glycosylated proteins and the ones that will secrete it from cell. So if somehow calreticulin and calnexin expressions are altered, or they are mutated, or something is not going on in ER lumen in terms of these proteins. So ER decide to abolish the protein folding mission and try to degrade these misfolded, unfolded proteins. But if the stress level is too high for the cell, for the ER, for endoplasmic reticulum, then endoplasmic reticulum starts a response, a very special response called unfolded protein response, and these unfolded protein response, or UPR, is very important cell process to decide the cell fate actually in a near future. So if in the ER lumen there is a folding problem, so in the ER membrane there's three famous proteins conducting this UPR response: ETF6, PERK, and IRA1. So these proteins actually start special signaling pathways. And all these pathways actually target two important things. First one is to start and actually elevate chaperone protein expression in order to fold properly all the proteins. So if it's not the case, cell can initiate autophagy response, which is that I will mention one or two slides later. It's a very special process. Is actually the recycle process of the cell. So in order to degrade or to eliminate all the unfolded or misfolded proteins. And finally, if the stress level, if the folding process cannot be properly done, therefore ER can initiate ER-associated death process or apoptotic process. So all these three protein signaling, cell signaling processes are very important after if ER gets stressed due to mis or unfolded protein accumulations. So how is apoptosis or autophagy is initiated? Especially by the ATF6 activation, there is some famous transcription factors like CHOP or ATF4, which are actually directly command to apoptotic proteins like BCL2, BCL-XL, or pro-apoptotic ones, PUMA, NOXA, in order to initiate apoptotic response, because we know that when BAX expression level go high, so mitochondria membrane potential is lost and apoptotic process can be started. Or when BCL2 or BCL-XL anti-apoptotic proteins are decreased, then your cell can initiate as well the apoptotic response in your cell. So for our scenario, When we compare these LNCAP and DU145 cells, in other words, androgen receptor expressing and non-expressing cells, you can see our calreticulin, calnexin levels go down, our stress proteins, PERK, ATF6 go up in these two cell lines. So we can say that EBR is a good candidate to initiate ER stress, actually. But while doing these analysis, we used IPA. IPA is the integrated systems created by QIAGEN. So it's a very good, a protein tool. And our IPA told us that your compound can act like tunicamycin, which is a very well-known ER stress inducer. So that's why we love very much IPA. You can determine all the actions, all the signaling pathways, all the proteins altered after your treatment. It's a very powerful tool to express to understand proteomic data. And we also confirmed our results that CHOP transcription factor is activated and apoptotic response is there after EBR treatment. So we finalized this part of the project saying that calreticulin expression is very important. After EBR treatment, it's downregulated. ER stress can be upregulated, causing CHOP activation and apoptosis finally. To be able to show that calreticulin is the main target of EBR, we ran another project, and in this project we used two approaches, basic approaches. One of them was the silencing calreticulin, the other one is overexpressed calreticulin, and see different cellular responses. Different cells, apart from prostate cancer cell lines, they actually give approximately same responses. And we again proved that calreticulin is the main target of EBR. So instead of trying to understand steroid hormones action, etc., we come up with a very different result. And finally, we also show that Box and Puma knockout cells, these proteins are very important in apoptotic regulation. So if they are knocked down, your cells can be more resistant to apoptotic stimuli. So even in these cells, EBR can initiate apoptotic response. And the other part of the project is to understand the autophagy processes, because we know that ER stress is very important to initiate autophagy process. Autophagy, as I said, is a very different cellular process, because it can initiate cell recycle mechanism. Cancer cells especially use autophagy to survive, and sometimes if the autophagy induction is chronic, cells undergo apoptosis in an autophagy dependent way. So in the cross section, we know that ER lumen, ER transmembrane proteins, this PERK, IRAVAN, and some IP3 receptor proteins in the ER membrane, can also initiate autophagy response via a mammalian target of rapamycin or BCL2 phosphorylation. So autophagy can also be initiated with ER stress. So during ER stress, as your unfolded, misfolded, or aged organelles, unfolded, misfolded proteins or aged organelles can be formed in a vesicle, can be degraded in this vesicle, and this vesicle actually is fused to lysosome in order to use lysosomal hydrolytic enzymes. And all these, as I said, proteins or organelles are degraded in this autophagal lysosome vesicle. So by doing this, cell actually can target two things. First of all, to degrade all the proteins and to restore energy level, and to use this micro component to build micro ones. Or the autophagy process can be initiated as well to initiate cell death actually. So depending on ER stress level, if the literature tells us that if the ER stress is acute, then autophagy process can be initiated. But if the prolonged ER stress is there in your cell, so cells can be die due to apoptotic induction. For this purpose, especially normal cell lines, not cancerous ones, they can cope with this ER stress very well. We saw that. For example, fetal human colon cells, mouse embryonic fibroblast cells, they can survive from EBR treatment due to autophagic induction for a while. And some of the cancer cells can also survive. For example, the aggressive colon tumor representing cell lines like SW480 or DLD1 cell can survive due to autophagic induction. Here you can see very nicely that an autophagic marker LC3 is upregulated due to EBR after 24 hours of application treatment. So these green dots represents LC3 protein, which is a marker of autophagy. Here again, you can see for another colon cancer cells. Another marker of autophagy is P63 protein degradation cleavage. So here in the control cells, untreated cells, you can see P62 expressions. After your treatment, all the P62 expression go down. So some of the cancer cells, again, as I said, can cope with this stress due to autophagy induction. And my PhD student also, he checked all the autophagy proteins expressions after 24 and 48 hours of treatment, and he showed that autophagy is there for some of the normal cells as well as some of the cancer cells, especially the very aggressive ones. So as a summary of our in vitro data, we show that EBR can induce apoptosis via ER stress, and after a very short term of EBR exposure, autophagy process can be triggered in cancer cells as well as in epithelial cells. But if your EBR treatment time go, for example, to 48 hours, so apoptotic induction can be initiated. So the second part of the project, of course, is to show EBR effect on colon cancer mouse models. For this purpose, actually, we try to understand, especially the aggressive tumors. For example, SW480, as I said, is a very aggressive colon cancer model, and we saw that autophagy process is there. So, and we try to understand in mouse model increased doses of EBR may be a therapeutic model for these aggressive colon tumors. So our data in 3D and 3D cell culture models and mouse models show that as long as we increase EBR concentration, we get less tumor size and tumor diameter. And we also try now to understand how much dose we can go up, because we know that EBR, due to its plant origin and steroid structure, high doses of steroid compound like this have very less effect in terms of metabolic syndrome or other diseases like they do not show any side effects actually to normal mice or other tumor-bearing mice. So we think that this concentration can be higher than 720 microgram per kilogram, because we know now that tumor size is decreased and tumor volume is decreased, and the health of the mice was okay, were okay. So it was very good result for us. Now, this part of the data now is unpublished, is prepared for publication. Now, we also show that tumor, normal mice or SW480 inoculated mice, they do not have any weight loss or any other health problems, and their serum colon cancer marker CEA is downregulated in a very nice way. We also show that their Ki67 or cytokeratin 18 levels, which are again tumor markers, are downregulated. And we also show that in these tumor samples, very important cell cycle proteins expressions go downregulated, so which is very nice results for us. So as a summary of the in vivo data, we show that EBR treatment decreases tumor volume and diameter at no significant effect of the overall health of mice, because when you dig the literature, there is no so at that time there was no good in vivo study or other patent or something like that to find a way for us. So that's why we choose to work with increased concentrations in mice. And we think that these, as I said, these concentrations can be increased to see maybe better results of in terms of volume diameter and volume decrease, tumor diameter decrease. Finally, the third part of the project was on EBR effect on Alzheimer's disease. So why we choose this path actually? Because we started to understand its action, EBR action on cancer cells. While we are doing our research, one of my master students he ran a immunoblotting. And he, without telling me, he looked for a GSK3 beta expression, which is an important marker of metastasis actually. So he tried to understand what's going on there in terms of metastasis, etc. But GSK3 beta is very important protein and very has actually very different roles. Normally, GSK3 beta, it stands for glycogen synthase kinase 3 beta, and it has role in glycogen metabolism, in metastasis, or in neurodegenerative diseases. And we suddenly thought that maybe GSK3 beta, which is very important molecule for Alzheimer's disease, can be a target of EBR. It really can be a target of EBR, and EBR maybe have some effect on Alzheimer's disease. So, and then while we are searching some papers, what's going on? Is there any relevant data about EBR and GSK3 protein? We saw this paper. This paper actually tells us that in Arabidopsis thaliana, brassinoid signaling can activate GSK3-like kinases, and the activation of GSK3-like kinases in mammalian cells can be, have various effects, especially GSK3 beta phosphorylation via serine 9 residue. Actually, it inactivates GSK3 beta. It's an inhibitory phosphorylation domain, and then we started. So this immunoblotting results actually belongs to my master students, belongs to Utku. Without telling me, he looked GSK3 beta in cancer cells, and he saw that after some time, cells can have a high expression of phosphorylation of serine 9. And then starting from this point, actually, we created some neuronal models, and we work with PC12 cell line. So PC12 cell line is a rat cell line and used for neural models. And we saw that when we treated this cell, the cell line with increasing concentrations of EBR, we got some, increase in GSK3 beta phosphorylation and some important molecule in neuronal survival like beta-catenin, and another cyclin-dependent kinase, CDK5, which is again very important in Alzheimer's disease progression. And therefore, we transfected ourselves with tau protein. Tau is very important for Alzheimer's disease progression, and we know that tau expression, tau phosphorylation is highly elevated in Alzheimer's disease patients. And therefore, after transfecting ourselves with tau, we checked its phosphorylation, and we saw here a very nice increase in tau phosphorylation. And then finally, we, created, not creation, but we made a model of Alzheimer's disease. And here you can see that cells treated with EBR after tau transfection has increased GSK3 beta phosphorylation, diminished Alzheimer marker enzyme, and here I will show you some data on C. elegans model, because C. elegans model is a very, either hard and easy to maintain in lab, and you have many different nice disease models if you use this nematodes. And then we started a project, an EU project, a COST project, and using these individuals. So we worked with very different types of Alzheimer models here. The ones with like GSK3 beta, CDK5. So very important molecules for Alzheimer's disease. So for example, here I will show you two different videos. So one of them is in the left part. So these are Alzheimer's disease representing individuals. So as you can see, they are dead. They do not move, and they are completely gone. But if you treat yourselves with this compound, after 15 days, they can still move around, and they are very happy. So these results actually made us very happy, and told us that we are in a very good way of showing the compound effect. And we run different experiments here. So using Alzheimer models of xenograft elegans, so we made some, again, movement experiments like chemotaxi experiments. So you basically put your C. elegans just in the middle of the petri dishes, and one part of the petri dishes is a chemoattractant, and the other part is not. There's the normal petri dish type, and you basically track your worms if they go to the attractant part. So the attractant for chemoattractant for these individuals is 100 millimolars of sodium chloride. So if they can move easily, so in these models of C. elegans for Alzheimer's disease, the tau protein and the AB42, which is the biomarkers for Alzheimer's disease, these proteins can accumulate in their muscles and they cannot move. If your treatment, target these proteins so they can move. So when you put your worms in the middle, so you tracked your worms. So what do you expect? Alzheimer models cannot move to the chemoattractant, or they are, go maybe the opposite part. But the treated ones, so we hypothesize they can move to the chemoattractant in a very easy way. And what we saw was, according to compared to transfection control, Alzheimer's disease individuals, they can move easily. So we tried this experiment in the presence of EBR or some CDK5 inhibitors like roscovitin. Because we know that CDK5 levels are very high in Alzheimer's disease patients, and EBR actually can, are more potent than roscovitin, CDK5 inhibitor in this case. And we also show that in these individuals in C. elegans model, again, Alzheimer's disease models, individuals, GSK3 beta phosphorylation is upregulated, and some of the analogs of Alzheimer's disease biomarker like BAGE expression, the analog of BAGE, downregulated in this expression in the presence of EBR. And we again show that in vitro models they have a nice upregulation of GSK3 beta serine 9 expression, CDK5 levels again it's decreased, and GSK3 beta expressions again they are downregulated, which are very good results for us. So this part of the study is now PCT pending, the international patent pending. And the first part, again, we got our national patent. So we can now state that EBR is a good candidate for GSK3 beta inhibition and can be used in Alzheimer's disease patients. So finally, our take-home messages from all these three parts of the study is EBR is an apoptotic inducer in cancer cells by causing ER stress. And normal epithelial cells or aggressive cancer models can cope with EBR-induced ER stress via autophagy. However, I forgot to say that in Alzheimer's disease models, we use a very low dose of EBR, so it doesn't have any side effects or so on. So low doses of EBR increases GSK3 beta inhibitor phosphorylation, which is a target of Alzheimer's disease. So when you search in the clinical trials or FDA approved some other drugs, you can see a GSK3 beta is a target for Alzheimer's disease research. And we also show that EBR can inhibit another Alzheimer's disease biomarkers, amyloid beta 42 accumulation, and allow, as I show you two different videos, C. elegans models can move again, so which is very exciting a moment for us. So in the near future, we try to check these results of AD, Alzheimer's disease, in vivo mouse model as well, and also we constructed some experiments, some projects to mimic a blood-brain barrier model in vitro model, and as well as in vivo experimental setup to understand EBR can pass through these cell membranes, because we know that some steroid-like molecules like dexamethasone are very much used in clinical, like in glioblastoma multiforme patients or some other invasive methods like brain surgeries, etc. They use dexamethasone, and dexamethasone is very much similar like EBR. So we hypothesize that EBR can easily pass, like dexamethasone, this blood-brain barrier. And that's why we started to construct these models in vitro and in vivo projects. So lastly, I would like to thank my collaborators, Elif Damla Arısan, which is a member of GTU as well, Ajda Çoker, and our United States project members, Professor Merali and Professor Barrero, and my PhD students Kaan and Leyla, and my alumni Utku, Tuba, Berna, Didem. They made a great job in understand GSK3 beta and C. elegans culture techniques, etc. And my master's students again, Eren. And lastly, I would like to thank Professor Barış Binay for inviting me today. And this was the last slide of my talk. Thank you very much for your attention.
Thank you very much for your nice presentation. If you have any questions, we can take right now. Any questions?
So maybe I can say one of my other future plan. For example, I also think that these plant steroid molecules are actually they have very potential to be as an antiviral candidates as well. For this purpose, one of my PhD students now, she's running all these in silico experiments to understand if EBR has affinity to, for example, SARS-CoV-2 proteins like spike proteins, etc. In collaboration with one of your Gebze Technical University members, we now set some experimental setup, Professor Mehmet Özbil. So we made some plans to check with in silico experiments if this compound have some potential to inhibit SARS-CoV-2 as well. So we think that due to its steroid structure and natural compound structure, it has many potential applications in terms of anti-cancer therapies, antiviral therapies, and anti-neurodegenerative disease therapies. So we continue to work with EU projects and TÜBİTAK projects and try to understand how can we can go far actually. So if you have some questions, I can answer. If not, this is the end of my topic and career and talk.
If you don't have any questions, we can end this webinar.
Okay, thank you very much.
Thank you very much for joining our webinar, and see you in the next webinars.
Thank you. See you.
Bye.
Bye-bye.